Axel, for your kind invitation. It's a pleasure to have the opportunity to share these new results with all of you. And I would also like to say thank you due to the great initiative with this series of seminars. So today I would like to talk to you about behavior and physiology of the forager honeybee, mechanisms that enable the specialization and the coordination of pollen collection. First of all, I would like to say that this talk will be organized around two main questions. The first is, what are the differences between pollen and nectar foragers that contribute to foraging specialization? And the second part, we will talk, or we will address, how do colonies select pollen according to its composition, or let's say quality. So, as you might know, division of labor is a feature of social efficiently. Division of labor enables different activities to be performed simultaneously by a group of specialized individuals of the working castes. In many social insects, division of labor correlates with the age of the worker, and this is the case of the honeybee, in which workers progress from in-hive task to perform tasks outside the nest, outside the hive, as they grow older. So at early ages, workers in cells take care of the brood and the queen. At middle ages, they become food processors. Afterwards, they are guards. By their third week of life, they go out for foraging resources. So, it is believed that workers differ in their behavioral responses because they perceive task-related stimulus differently. A good example are the guards, who are the first group to display defensive behaviors when, for example, the colony or the entrance of the colony is threatened. And this is mainly because they are more likely or prone to extend their sting when exposed to aversive stimulus or even the alarm pheromone. In the foraging context, as you might imagine, nectar and pollen stimulus are very relevant for the behavioral response. Pollen and nectar are the two main food sources for honeybee colonies, nectar providing energy, carbohydrate, while pollen provides lipids, minerals, but mainly proteins. And in the honeybees, pollen and nectar foraging are carried out by different groups of bees. There are bees specialized in nectar foraging and bees specialized in collecting pollen. There are fundamental differences between both activities. Nectar foragers lick liquid food at the flower. It ingests the liquid food and store it in the crop, in their crop. They can also use sugars in nectar to fuel their activities, their foraging flight, for example. But on the other hand, pollen foragers rarely consume pollen at the flowers, at the sources. Indeed, they transport pollen to the hive externally in their hind legs, in some specialized structures called corbicula. This foraging specialization, bees or foragers collecting nectar and foragers collecting pollen, differ in how they perceive nectar compounds such as the sucrose. Sucrose sensitivity can be evaluated by means of the proboscis extension reflex. The proboscis extension reflex is an innate response elicited after we touch the antennas of the bee with enough concentrated sugar solution. Growth sensitivity can be then estimated by stimulating the antennas of the bee with solutions of increasing concentrations and by determining which of these solutions, which concentration is concentrated enough to elicit the extension of the proboscis. Here in this example, we have that these bees needs a very highly concentrated solution to extend their proboscis. Then it is less sensitive than this other bee. I don't know if you can see my point, but okay.
Yes, we can see it.
Less sensitive than this other bee who requires or can extend their proboscis even with a intermediate or low sucrose concentration. This is a figure published by Scheiner who show what we were talking about, differences between sensitivity among both forager types. Here we have the percentage of bees showing proboscis extension response, and here we have the series of increasing concentration of sucrose. And here we can clearly see that pollen foragers are much responsive to a broad spectrum of sucrose concentration than nectar foragers. Yeah, then pollen foragers are more sensitive than nectar foragers. It's worth to mention here that these experiments and other similar experiments were performed with foragers captured at the entrance of the hive. It means that foragers were returning or at the end of their foraging visits. Then the fact that nectar foragers are present low sucrose sensitivity might be adaptive as enable nectar foragers to concentrate or to focus on collecting nectar sources that provide a high reward, and this is good for the colony because the energy gain is higher. However, it's not so well understood why. Sorry. Okay. Here you go. I was saying that it's not completely clear why do highly sensitive foragers like pollen collectors, pollen foragers, prefer to gather pollen instead of nectar. And here comes our first hypothesis, that is that probably we are missing some information, and foragers arriving at the pollen source could be less sensitive than we previously thought. So here comes our first question, that is, are there differences in sucrose sensitivity between foragers arriving or departing from their sources. And this is an experiment. I will show you experiment led by Emilia Moreno. She's a PhD student in our lab, and she trained the bees, initiated this experiment, training the bees to a foraging station. The foraging station offered two feeders, a pollen feeder and a sucrose solution feeder. Bees arriving at both feeders were captured, and also Emilia captured bees soon after they leave the feeder. As a consequence, we get 4 different groups: pollen foragers arriving, pollen foragers leaving the feeder, nectar foragers arriving, and nectar foragers leaving the foraging station. She harnessed the bees in those metal tubes, leaving the antennae and mouth part of the bees free for the stimulation, and she proceeded to make this protocol to estimate the sucrose sensitivities of this group of bees. During this procedure, it's important not to feed the bees, yes, because this would change the motivation or the satiation state, and that may change their response. And in between the solutions that we offer, always bees were offered water. If the bees were thirsty, they were allowed to drink in order not to mix up responses due to water from those triggered by the sucrose. Then here we can see our first results. These are for bees, pollen and nectar bees leaving the feeder. As you can see here, we have the proportion of proboscis extension response. Here we have the 7 concentrations that we offer during the experiment. And as I already consistently with the results I show you from China, there were significant, very important differences between pollen and nectar foragers. Consistently with previous results, pollen foragers were much more responsive than nectar foragers, than nectar foragers. But then, when we analyze the bees arriving at the feeders, we still found significant differences between both curves, but interestingly, the results were inverted. Now you can see that pollen foragers arriving at the feeder were less responsive than nectar foragers. Then there are differences in sucrose sensitivity between foragers arriving and departing from feeders, and then this might let us speculate that pollen foragers at the beginning of their foraging visit would not be so attracted to the nectar as we previously thought. Another very important aspect of foraging behavior is learning of environmental cues that predict food resources. When the honeybee or when a foragers discover a flower or a floral patch, she can associate neutral cues from the flower, such as, let's say, the odor or the flower color, or even their shape, with the reward that the flower provides. As long as the honeybee continues visiting this floral patch repeated other rewarded associations turns into memories that improve foraging efficiency. For example, by guiding the foragers towards the learned odor, triggering a landing response on the flower, and even eliciting discrete behaviors such as the extension of the proboscis when the bee approached to the flower and perceived the learned odor. The conditions and parameters in which the bees learn can be recreated in the laboratory, mainly by means of the conditioning of the proboscis extension response, normally in the olfactory modality, conditioning with an odor. The conditioning involves at least two stimuli. The neutral stimuli, here it's represented by the odor. It is neutral because when presented before conditioning, it did not elicit any response on the bee. And the other stimuli is the reward. In this case, and in most cases for the proboscis extension conditioning, was the sucrose solution. Then, during conditioning, the association that between the odor and the sucrose solution enabled the bee to establish an association that after this pairing occur may lead the bee to elicit their proboscis just upon the odor presentation. It means that after several conditioning trials, the odor predict the oncoming reward. Yeah? Here, I would like to show you a video to see how this conditioning occurs in real life. I think it's working. No. No, it says it cannot play media. Sorry. Okay. Probably later I can show you the video, not directly in the presentation, but afterwards. The idea is that in this video, we will show how the odor is delivered upon the antennas of the bee, and during the odor presentation that normally it is presented during 6 or 7 seconds, we offer together with the odor the sucrose on the antenna, and here we have this pairing that enable that after 3 or 4 conditioning, the odor alone controls the extension of the proboscis. So, most of these studies investigating learning and memory use reward, sugar reward for the conditioning, but only a few studies have investigated the function of pollen as a reinforcement of learning. Indeed, bees extend their proboscis when their antennae are stimulated with hand-collected or even bee-collected pollen. However, this response is not very stable, and in most cases, bees stop responding after a few pollen presentations. So in the laboratory, we developed an alternative procedure that we called the double reinforcement, in which we condition the bee using simultaneously sucrose solution applied on the antennas of the bee, pollen, normally hand-collected pollen, on the left tarsus of the bee. This double reinforcement, as before, enable to establish an association between the odor and both rewards, and we are confident that with appropriate controls, this procedure allowed us to estimate the contribution of pollen during learning. So, using this procedure, we to the second question, that is, do pollen foragers perform better than nectar foragers during conditioning using pollen as this double reward? Here, this experiment was also performed by Emilia Moreno. She captured bees, pollen foragers, and nectar foragers arriving at the foraging station, and she harnessed them and initiated a conditioning that consisted in 5 acquisition trials. This is to establish 5 associations between the odor. Here we get the odor, and simultaneously we present the sucrose on the antenna and pollen on the tarsus. This procedure, we call it the paired procedure, as pollen was paired with the odor. And we also perform a second procedure, which we call unpaired procedure, in which the association during conditioning was established between the odor and the sucrose. Pollen was also presented, but afterwards. It was not paired, so it does not contribute to memory formation. Another parameter to complete the conditioning was testing memory extinction. This memory extinction was tested through 4 extinction trials in which the bee was exposed just to the learned odor to see how well the memories established during conditioning can be retained. So here are our first results, or results from this part. We have this, we show you first the procedure, meaning that the bees were established the association between the odor and the sucrose solution, because pollen was unpaired. And we can see that during acquisition, both arriving foragers from pollen or nectar type perform very similarly. Here we have the percentage of bees extending the proboscis during 5 consecutive learning trials. So we can see that there are no differences between bees, not during the acquisition of the olfactory information, neither when we test memory extinction. Both memories extinguished similarly. Now, when we move to the paired procedure, in which now the odor was rewarded by sucrose solution plus pollen, we can see, or we can detect, differences between both groups. With pollen foragers be performing better, learning faster probably, than nectar foragers, and also retaining their memories better than nectar foragers. As a conclusion, we might say that pollen foragers learn odors reinforced with this double reinforcement, sugar plus pollen, better than nectar foragers. Using the same experimental procedure, we also tested, now working just within the pollen foraging subcaste, whether or not pollen foragers that arrive at the sources perform better during learning than those pollen foragers leaving. Here we have the results for these pollen foragers departing from the feeder, and here you might take into account that we are comparing performances obtained by the unpaired procedure. Here, those are blacks. It means that the association was established between the odor and the sucrose alone. Again, the other curves with this document. Here, so at departure, there were no differences between the learning established with these two procedures, nor during acquisition, neither during the extinction. We move to these pollen foragers arriving at the food source. We found significant differences. Pollen foragers that were trained or conditioned with the double reinforcement that were just rewarded with sugar, meaning that memory is established just with sugar, extinguished faster than those obtained with the double reward. This suggests that the contribution of pollen as reinforcement during learning is higher in this arriving at the feeder than this departing the feeder. So, as conclusion for this first part of the talk, we obtained that pollen forager sensitivity to sucrose is lower at the beginning than at the foraging, at the end of the foraging bit, and this might imply that the bees are less attracted to nectar sources at the beginning. In the last experiment I showed you, we observed that contribution of pollen as a reward is relevant at the beginning of the foraging visit, and might indicate then that pollen foragers arriving are more sensitive to pollen than when they leave the station. And from comparison between foragers type, we get that pollen foragers perform better than nectar foragers when conditioned with this double reinforcement, a fact that might contribute to foraging specialization. Okay, now let's move to the second part of the talk. And the main question here is, how do colonies select pollen? Pollen composition greatly varies according to the floral origin. Pollen can present a phagostimulant component, but also toxic or deterrent compounds that might lead to some rejection during foraging. We mentioned that foragers it is believed that foragers rarely consume fresh pollen at the sources. Indeed, pollen is transported externally in their corpicula, and I already showed you that foragers learn with pollen through chemotactile stimulation. It means digestive stimulation. So it led us speculate that pollen assessment by foragers occur without pollen ingestion. Then we argue that foragers select pollen based on their innate or even learned preferences for pollen-based cues, such as intensity of pollen odors, their color, their texture, and even the taste of nutritional or non-nutritional compounds. Once the bee or the foragers return to the nest, the pollen is highly or greatly manipulated and processed by hive bees, mainly nurse bees of 3 to 8 days of age are in charge of processing this pollen. They are the ones who consume most of pollen from the reserves. They digest pollen and also, or let's say, to produce a jelly that is rich in protein, which is used to feed the larvae, and even it is shared to all the colony members. And then we hypothesize that pollen, even when pollen foragers are sensitive to certain components in pollen, they might be able to learn about pollen composition or certain components of pollen inside the nest after pollen was processed and handled by these hive bees, like nurse bees. So here I will present you a series of experiments, experiments performed by Rocío Lajad. She is a PhD student of our group, and she led this experiment. The first question was, to what extent foragers make decisions based on pollen composition, and whether foragers can adjust their foraging preferences after they have the opportunity to experience pollen inside the hive. So what Rocío did is to confine colonies inside these big flying cages. Here you can see at one of the end of the cage the colony, and she trained the bees from the colony to a foraging station that at a certain time it could offer 2 different feeders, 2 pollen feeders that were used during this dual choice test. One of the feeders offered, let's say, pollen A, and the other feeder offered pollen B. These pollen A and B were monofloral bee-collected pollens. And the idea was to compare preferences, foraging preferences for pollen A and pollen B when one of the pollen, namely pollen A, was adulterated with a deterrent compound. In this experiment, I will show you that we adulterate pollen with amygdalin. Amygdalin is a deterrent substance that has been shown to induce malaise experience after it was diluted in a sugar solution and offered to the bees. And it is a compound that can be found in pollen from almond trees. Just a second. Come on. Okay. Oh. Sorry. There is some. I'm opening the presentation again because it collapsed.
No worries.
Sorry. I don't know if you want to make some comments or while we wait until the computer react. Now it seems that it's coming.
Okay.
Just 1 second. I will share the screen again. Can you see the presentation again?
Yes, it's back.
Perfect. Okay, sorry for the delay. So let's move quickly to this first experiment performed by Rocio. In this first experiment, we want just to see how amygdalin, this deterrent that need to be ingested in order to induce malaise experience, affect foraging preferences or initial preferences of foragers. So we have, as I said, a flying cage with a colony. So we have bees from these colonies making these dual choices between both feeders offering pollen A and B, and in one of the treatments, pollen A was adulterated with amygdalin, 0.1 molar, and these preferences, initial preferences, were contrasted with other colonies from bees from other colonies that were have the opportunity to choose between pollen A and B when both pollen A and B were unadulterated. So this is what we get. Controls, colonies. Here, first of all, I would like to mention here what we got here. Those are foragers choosing pollen A over total number of foragers using pollen A plus B. Here we have the two treatments, control, which were the opportunity to forage between unadulterated pollens, and the treatment called amygdalin. That is why, or because pollen A was adulterated with amygdalin, then foraging preferences of both group of colonies were similar. There are no differences, indicated that these initial preferences were irrespective of the deterrent addition. That is to say that foragers were insensitive to the amygdalin aggregation. Then we move to a second experiment, which is a bit more complicated, but still quite intuitive. We have pollen A that instead of being offered at the feeder, it was offered adulterated inside the hive for all the bees of the hive. Yeah, this pollen was offered inside the hive for 2 consecutive days, and we start measuring on day 1 the initial preferences. We get the treatment, and on day 4 of the experiment, we tested again preferences for both unadulterated pollen. So while we have the treatment in which pollen A was offered inside the hive adulterated with amygdalin, we have control group in which pollen A was offered inside the hive without any addition. It means it was not adulterated. And in a third experiment, we follow the same reasoning, but here we let the honeybees or the foragers to introduce naturally the adulterated pollen into the hive. Yeah? The idea was the same, that then during these 48 hours, the bees have the opportunity to introduce adulterated pollen into the hive. And again, at day 4, we measure preferences for both pollen, now unadulterated. Here, I will show you the results for experiment 2. It was repeated with 4 different colonies for the control, and for 4 different colonies in the amygdalin treatment. As you can see here, preferences of foragers were reduced in the amygdalin group. Yeah, here it's worth to mention that the initial preferences we measure at the beginning of the experiment was introduced into the statistical model as a covariate, yeah, in order to account for qualitative differences between preferences of different colonies. So we have just to compare here the preferences at day 4, at the end of the experiment, after the bees experience or have the opportunity to learn the pollen, adulterated pollen, unadulterated pollen inside the hive. So in experiment 3, where the difference was that the bees were those who introduced pollen, again, we found significant differences, and foragers somehow reject pollen A in their testing events. So from these 2 experiments, we might say that foragers learned to avoid pollen A after it was experienced inside the hive. The last experiment, experiment 4, followed the similar schedule, but here we capture these after they show their initial preferences. We confine them in these small cages. Those were all foragers that here after they revealed their preferences, they were marked with a colored dot. They were then confined in these cages, and according to the treatment, they could receive adulterate pollen A or unadulterated pollen A in the control group. They were introduced into the hive, and this condition was maintained for 48 hours. Then they were released, and on day 4, they were tested again against unadulterated pollen A and B. Here, we found no differences, meaning that foraging preferences did not change after these individuals were confined in strength contact with the adulterated pollen. What might say about these results? Okay, we argued that information about pollen composition is somehow transferred inside the hive, that the foragers need to be in the social context to gain or be able to learn about pollen composition, and that might be mediated, or these transfers of communication might be mediated by in-hive bees. In particular, the nurse bee are good candidates for assessing, learning, and remembering information about pollen composition. Those are the main members of the colony who handle processed pollen, as we already mentioned. So the next and last question of the talk is whether or not nurse bees could learn specific cues according to their composition or quality. Rocio performed this experiment using bees reared in the laboratory to get a comb with silly brood. After immersion, the bees were confined in these golden cages in group of about 80 bees of the same age. These cages were fed with sugar solution, and cages also receive 2 monofloral pollens, that were irritated and offered in these tubes, which were graduated, meaning that we could measure or calculate how much pollen the bees consume. So during the first 2 days of the experiment, we calculate consumption preferences of the cage. We move the training phase on day 3 and 4, and here again, pollen A, one of those monofloral pollens, were adulterated with different substances. We get 2 treatments using amygdalin, other treatment using quinine. Both are acting as deterrent. We also have linoleic acid added to pollen A as a phagostimulant, and we also keep a cage, a control, in which during training both pollens were offered unadulterated. On day 5 and 6 of the experiment, we move to the testing phase. Again, pollen A and B were offered unadulterated, and we measure the consumption preferences of the cages. Here we can see the results. As in other experiments, here we have the consumption of pollen A against over the consumption of pollen A plus B. Here in this axis we have the 4 treatments. You might notice that controls prefer pollen A over pollen B, but these preferences was not really modified, was not significant when we added linoleic acid to pollen A. But consumption of pollen A was reduced. There were significant differences when we use amygdalin or quinine to adulterate the pollen. Here again, the initial the consumption preferences obtained during the initial phase were used as covariate to account for differences in pollen consumption among cases. And here you can see the number of replicates, number of cases that we have in each treatment. Then, from this experiment, we might get that nurse-aged bees associate pollen cues with distasteful or malaise experience that okay. The conclusions and probably some discussion of this second part would be that foragers are relatively insensitive to certain compounds, but they were able to modify their preferences for those pollens if they were experienced inside the hive, likely mediated by in-hive or nurse bees. And from this experiment, we might get that young bees perceive and learn pollen characteristics, and we suggest that they might be involved in mediate this pollen selection. The question that we need to address now is what are the mechanisms that enable such transfer of information between in-hive bees and foragers. This is something that we are working on. Okay, I think that it was enough. Thank you very much for your kind attention. And here I would like to give you an email and the web page of our institute. Or say thank you to these 3 financial institutions. Thank you very much.
Thank you, Andrés, for sharing these very interesting results. Just a reminder that if you would like to ask a question, feel free to raise your virtual hand and ask Andrés directly. But I saw that we also had a couple of questions in the chat, I think, Axel.
Okay, yeah, so there are 2 questions in the chat, Andrés. So the first one is, and that is with respect to the first experiment, did you control for the odor of the pollen? Do you think pollen was acting as the US or an additional component of the CS? Andrés?
Yes, indeed. I would like to move to this part. Sorry. Here.
I think so, yeah.
Yeah.
Maybe you also say which odor you used for the conditioning.
In this case, we use linalool. it was quite common during conditioning. It was well, it was salient all over, and yes, the question is valid, and even I think that the main argue is that we here also present the odor even during the trials. Of course, it was unpaired, and in this case, if there is any effect of the odor during conditioning, it might be also affect or interfere during this procedure. And that's why we try to keep all the trials, all the learning trials, with the 3 stimulus offered within each 1 minute approximately. But then if there is some association between sugar and pollen odor, okay, then we might have see that this contribution is not the same here and here, because here the association, or the probably association between the pollen odor and nectar would not contribute to the response we observed.
Yeah, I guess I just wondered if having some odor from the pollen helps those pollen foragers to learn the unconditioned stimulus more easily, because there's an overlap between the presentation of the sucrose and the pollen. I suppose if you had another treatment where you just presented the pollen but didn't allow the bees to kind of make contact with it in the unpaired condition or in the paired condition, that might be interesting as well, to see which kind of component of the pollen reward is giving this additional learning.
Yes, you're right. And indeed, we have at least 3 or 4 more experiments supporting this evidence. You may find these experiments in this paper. This is a paper from Neri, Moreno, and Arenas in 2020 in Experimental Biology, Journal of Experimental Biology. And there we also stimulate, for example, the hind legs of the hind legs of bee. Also, we stimulate the antenna. We try with hand-collected pollen, with bee-collected pollen. There you will find some other evidence that also suggests that pollen foragers are more sensitive, or at least are more excited by pollen than nectar foragers. Yes?
Yeah, sure. Thank you.
Okay,
Okay,
Andrés, I have a question. So does pollen stimulation of the antennae or the legs, the tarsus, elicit the extension of the proboscis?
When we use bee-collected pollen, that we know that during collection, bee glue, stick pollen grain with nectar addition. So bee-collected pollen might contain sugar, might contain nectar, and it is a good stimuli that induce extension of the proboscis. Hand-collected pollen might elicit extension, the proboscis extension, but it's not so stable. And that's why we managed to modify a bit this procedure in order to have bees that continue responding to pollen, or at least pollen presentation, sugar plus pollen, along at least 4 or 5 trials. One of the main problems with just using pollen on the antennae is that pollen gets stuck in the antennae, and then somehow the bee stopped responding. So it's a mixture between, I don't know, somehow it's a methodological problem, but on the other hand, we found better results when we managed to give this double reinforcement.
Okay, let me ask you one other question, then I'm reading the questions again. So do you think that amino acids solved in water, and then you do a stimulation of the antennae, whether that would lead to the extension of the proboscis?
A recent study realized or discovered that the bees were able to assess amino acid content in a solution, meaning that they are sensitive to this kind of stimuli. However, an amino acid solution do not trigger extension of the proboscis.
Thanks. Okay, so then here's a question of Tapovan Farm. What is the percentage of nectar versus pollen foragers, and how does this vary as the strength of the colony increases, so seasons, queenlessness, and diseases?
I don't know if I completely understood the question.
It was kind of like, what is the proportion of nectar and pollen foragers in a colony, and whether that proportion changes with respect to season or other kind of like colony states, like diseases.
Okay, the proportion can vary according to the season, of course. It is in a strength relationship with the amount of brood. Then at the beginning of the spring or at the beginning of the foraging season, the brood increase, then pollen foragers are more and more frequently found at the entrance. The proportion is about, I would say, 65% nectar and 35 pollen foragers, but this number may change according to the season and depending on the amount of brood that the colony has.
Okay, then there's a statement from the same listener, but I cannot really make sense of this. So this may not look relevant to the current experiments, but here are assuming that the colony is healthy and has good number of bees and sufficient food. I'm not sure. Maybe Tapovan, I mean, maybe you can raise your hand and we can allow you, you can ask the question to Andrés.
Yeah, I was just giving a background on why this question was raised. Basically, when the colony is under stress or when the food proportion is not sufficient, I think the experiments that we are seeing in the this set of results might be skewed. So that's why I was wondering if there was some graph or something like that, so that we can know how valid are these results.
Okay, thank you for your comment. Yes, yeah, I have two things to say in this regard. For learning experiments, we used bees that were previously trained or visit a feeder. So in most of these cases, we do not consider from which colony the bees come. So they were just identified as pollen or nectar foragers. Probably those groups come from very different places, from different colonies. But one thing that is important, that it's not easy to train bees to a pollen feeder, for example, when there are very good opportunities or very good food sources in nature. So we succeed in training bees to these feeders mainly by the end of the season, at the end of the summer or early autumn. So I will guess that yes, that all the colonies we use are much more or less in the similar state. And then when we move to the experiments in flying cages, we also found something that is interesting, that not all the colonies present similar foraging or even consumption preferences for the same pair of monofloral pollen, meaning that the background information or the experience or the, I don't know, the reserves of the nest influence which are the sources that are more appropriate for the colony. And that's why in all our experiments, we include these initial preferences, this starting point at which we measure, okay, those are the initial preferences of this colony. We have to take into consideration these initial preferences, calculate if there are or not differences after treatment. And that's why we always introduce these initial preferences in the model as covariate. I don't know if I answer a bit your question, but I hope yes.
Okay, Andrés, there is a question by Chin Chao. Chin, you can unmute your mic.
Hi, Andrés. Thanks for the interesting talk. I found your results are very exciting. For now, I have one question. I'm particularly interested in some of your early slides that you show today. So for the part where you show the changes, basically in sucrose sensitivity before and after the pollen foragers collect pollen. I think this is the first time I saw similar results, but somehow I think the results are somewhat counterintuitive, because I have always thought that the differences in sucrose sensitivity between pollen versus nectar foragers is a key for them to determine what they're going to be collecting. But as you showed in your slides, they have actually much reduced differences in the sensitivity before they collect pollen. I was wondering if you could provide more about the biology behind this. Wouldn't it make more sense if they have larger differences before they collect?
Yes, I will go to the presentation because probably the order in which I present these results start with the bees departing. But I completely agree with your comments, so I will just show you this. And here, if you can see, those are bees leaving the feeder. It means that they were at the end of their foraging visit, and in this were more sensitive than nectar foragers. This is in agreement with studies shown by Scheiner and other authors. Yeah, you see that the curves are very similar, with pollen foragers being more sensitive than nectar foragers. And in this experiment, foragers were captured at the entrance. And here, similarly, we captured the bees not at the entrance, but when they were leaving from the feeder. So it's almost the same internal condition of the bee. Those differences might be due because this nectar forager is full of nectar, probably satiated, and this pollen forager is hungry. Yeah, even when she arrived to the pollen feeder with some nectar in their crop, she used this nectar to stick and to gather pollen. So probably this is a condition in which the bee needs energy, and that's why it's very sensitive to sucrose solution. But at arrival, yeah, this is when he arrives to the pollen feeder, pollen foragers is not sensitive or not so sensitive to sugar than when leaving. And this is because probably they have some fuel, still have some nectar in their crop. Their hunger state is not so strong, then she prefer, or at least is not so attracted to nectar sources. Here, those bees, probably nectar foragers, come without any nectar in their crop. Probably they are a bit more motivated to get carbohydrates, and then they are a bit more sensitive than this group. I think that this is that we were talking about the same. I think that we agree in this. I don't know if you want to request.
No, I think this is a very important discovery, because it may change, completely change some of our theories. But I still need some time to digest these results. I think you mentioned that the manuscript is submitted, right?
And another thing that might complete this interpretation are results from here, in which on the one hand we observe that these pollen foragers are less sensitive to sucrose at arriving at the food source. So in a flower that offers both nectar and pollen, the bee might reject or avoid collecting nectar and focus on pollen. And here, while the bee pollen foragers arrive at the feeder, we saw that they were more somehow interested in responding or in learning information when pollen is involved in this conditioning, in this association, than when departure, meaning that at the beginning of the foraging boat, the forager is also more sensitive to pollen. Yeah, that's why it learns better. So I think that both information, less sensitivity to sucrose at arrival, but high sensitivity to pollen at arrival, makes the mixture that allowed the foragers to direct their preferences toward pollen.
All right, thank you. Thank you very much.
You're welcome.
Okay, Andrés, I have a question to the experiment before and leaving the feeder. Can you go back to that slide?
Yes, sorry. Perception?
The change in the sensitivity before and after, this one, yeah. So actually kind of like, so you collected the bees that were arriving at the feeder, and then you collected bees that were leaving the feeder, right?
Yes.
So actually arriving would be before leaving. And the way you kind of like made the logic was you were starting with leaving. So if you think about that, the change in the sensitivity is a result of being at the feeder, right? And so what I want to say is that the hive doesn't come into account here, in my opinion, but that one can discuss later. So the interesting thing was actually what I wanted to ask you. It looks like that the change in the sucrose sensitivity in nectar and pollen foragers is in opposite direction. Or is it that there is no change? I mean, do you think it's in opposite direction?
Yes, I think. Sorry that I start with the parting foragers, because this result is consistent with previous evidence, which just analyze the parting bees. But of course, the correct order is the other way around.
I mean, I'm saying this because I don't know whether you know that I did a study on the change in neuropeptides between arriving and leaving the feeder. And kind of like, I actually have to look into my publication again. And there might be even also opposite changes in specific neuropeptides. And then one could check whether these neuropeptides might have an effect and might have different effects on the two pollen and nectar feeders. So I just want to make that statement. I can kind of check it and then I can send you an email.
I'm aware of your publication. I have not in mind exactly the pictures now and the figures, but
me neither. And so I will check it and then communicate with you.
What I can say about this difference in this arriving and departing, it was a experiment we performed with Christoph, indeed, 2 years ago, and it was not designed to check for differences at the level of octopamine receptor in bees arriving and departing, but I think that it's somehow an insight into these differences. In this experiment, we analyze gene expression in different of octopamine receptors and thiamine receptors in different parts of the bee brain. In this case, the focal bees were nectar and pollen foragers, arriving at a feeder. Yeah, quite similar to what I already shown in the experiment. And here we found differences for the whole brain in one. Sorry, I have to move this. Receptor octopamine, receptor 1, alpha 1. And a different experiment, similar but a bit different, was performed with bees, known age bees that were introduced into the hive. We wait about 2 or 3 weeks and recaptured them when they were returning, either loaded with pollen or nectar. And then we measure again gene expression, and we found that have been detected in foragers arriving at the food sources were not present in bees returning or arriving to the hive. And the differences we found in this condition with bees returning to the hive was on a thiamine receptor 1. So again, this is not strictly, it's not comparable, because here we use non-aged bees, we control the age of the bees, and here we could not control the age of the bees, but at least these differences in which receptors are more expressed in the brain of nectar of pollen foragers might give us an idea of what might going on regarding differences in sucrose sensitivity.
Okay, just one comment. What they say, there are about 100 neuropeptides compared just to a handful of like 12 biogenic amines. So how to say, and of course, peptide research hasn't been kind of started yet in honeybees, right? But kind of like fast changes can also be done by neuropeptides. But okay, that is a general comment. So I will get back to you with an email. So now there's a hand raised by Margarita Olava. Margarita? We cannot hear you, Margarita. Margarita, you can unmute your mic. So, okay.
Okay. But she wrote her questions as well, so I can maybe read the first one. And it's a question concerning amygdalin. Does natural almond pollen cause the same adverse effect as other pollen adulterated with amygdalin? And she's asking because this has implications for commercial pollination of almond orchards.
Yes, this is a very interesting question, and it is hard to answer, mainly because we have relative success in studying the effect of these pollens adulterated with amygdalin in a very controlled condition. As I show you, there were flying cages just to control both the bees belong all to the same colony, but also to avoid bees collecting other alternative sources. So our system was very close. I guess that this is part of it's the reason why in part we succeed. We try to replicate some kind of amygdalin adulteration in sunflower fields, yeah, trying to control or at least modify preferences of foragers in the field, and it was almost impossible. I mean, not impossible, but we do not succeed. Our results, our preferences were not modified in the field. This story is because I guess that in the almond orchard, probably the situation in which the bee collects alternative wild flora or whatever might make differences between a resource that probably it's not the best quality, or okay, it's not the best quality, but still it's good, and we have access to it, and we can use it. I guess that natural concentration of amygdalin in almond, it's not so high as we use here, so probably the effects are not so strong that the one we induce in our experiment.
Christoph, do you want to read the second point?
Yeah, so there's also a second question. Do you think your results would be different if you used the pollen hoarding strains as opposed to normal commercial bees?
Can you repeat me, please, Christoph? I couldn't get.
Do you think the results would be different if you would use the pollen hoarding strains that have been studied in some previous works, as opposed to normal commercial bees?
This experiment, I always mention pollen A and B, but you see that indeed pollen A were different pollens. We use, in this experiment, for example, we use 4 different pollens A, which then in the model was taken all together as part of the same factor. So I think that there are differences according to the pollen or the quality of the pollen we use. However, when you get the tendency tend to be similar in different pollen. I mean, one of the reasons we prefer to adulterate a pollen is just not to use commercial pollens that are considered to be high quality or low quality, because low or high quality is relative to the condition of the hive. It depends. A good quality pollen for one colony could be a less quality pollen in other colony, because it requires exactly the requirements of the hive. So we prefer not to use pollens considered of being, for example, with high protein content as good quality pollen, but just to keep 2 known pollens and modify the quality by the addition of some substances.
Okay. So
Then there was another question, which was which subspecies was used in your experiments?
This was Apis mellifera ligustica.
Yeah. I also am very sorry.
I still have a question.
Okay.
I'm raising. So I found it interesting that you said that there might be an interaction between nurse bees and foragers. I mean, I kind of like only know from the nectar forager that they are then the receiver bees. And so I don't know. So can you elaborate a little bit on how and when you think nurse bees and foragers would interact?
Yeah, yes, I can. Those are speculation because we made different preliminary experiments and we failed. So I still will tell you what we think that is going on. We think that the foragers might learn inside the hive based on interactions with nurses, or even learn the modifications that nurse introduce into the hive. For example, our reasoning is the following. If we observe that nurse bees have preferences for consume one pollen and reject other, this less quality, or let's say the adulterate pollen, would be less consumed by nurse bees, then it would start being accumulated inside the hive. It can somehow be accumulated inside the hive because nurse don't want to consume it. They know it's less quality, and then the foragers have the opportunity to learn in base of those pollens that remain inside the hive for, I don't know, 2, 3, 4 weeks. So this pollen also get older and less attractive. So somehow there might be cues, pollen-based cues, that foragers might learn because nurse bees modify the social context, the pollen available in the reserves based on their own experiences or assessment. This could be one mechanism, but we have to check it. We have to prove it.
Okay, let me ask you then one question. Do you think that colonies that are brought to these orchids, forgot the flower name, that are producing amygdalin, do you think that there is a reduced brood production in that colonies because kind of the nectar, the nurse bees are avoiding the pollen?
I don't know. This is a bit what I was trying to say before. When we are in an open field experiment, change and differences are always different, or such difference we found in flying case diluted. But yes, there might occur some kind of rejection, but we cannot completely consider if this difference, small differences, make a difference in the, let's say, development of the hive, because we don't know exactly what is the contribution of this rejection in the whole gathering.
But so it's interesting, that's my last comment, because we had in the last seminar, we had the question, so the food quality control might be at the level of the nurses and not on the foragers.
Yeah, I totally agree. We have, at least for pollen, which is a quite complicated resource or stimuli, I will argue that nurses are those who really select pollen by consuming or not consuming the most appropriate pollen. So then they give somehow of feedback to the foragers, which are due to their repertoire or their state, physiological state. They are not able to fully evaluate or assess pollen at the food sources, so they need somehow the feedback from in-hive bees to make the most appropriate selection or foraging.
Okay, thanks. Christoph, go ahead.
Well, I just also wanted to briefly comment that I'm quite amazed and also really excited about the findings that you presented from before and after leaving the feeder. Very often we just treat a forager as the bee that returns to the hive, and we compare it to all kinds of other things. And what your results highlight is that a foraging trip is a complex operation that consists of different phases and involves lots of different complex behaviors and probably motivational changes, and that maybe we have to rethink how we do experiments where we want to compare foragers with something else. Maybe we need to start catching bees not just when they come back, but also when they leave. And then we might find big differences. That was my take away message, that we have to be very careful what we call kind of a generic forager.
Okay, Christoph, sorry, kind of like that. I also have a comment. So there's a problem with the PER. So Jochen Erber and someone else did an experiment where they compared the sugar sensitivity of free-flying bees and kind of like then caught in the PER. And actually they showed that the PER does not reflect the sugar sensitivity in the free-flying bees visiting a feeder. I think that for me, this was always a work that was never really kind of taken seriously, or kind of like really kind of evaporated throughout the papers, because that is then kind of like a little bit questioning the sugar threshold in the PER, in restrained, in the restrained condition of the PER. Okay, now you have to have another closing word.
Any other questions? Last question, last opportunity.
So there's one from Natasha Rossi.
Oh, sorry, I
Andrés.
missed that.
No, it just came, I think. Hi, Andrés, I'm sorry if I missed this information because I arrived late, my bad. But do you know by which mechanism nurses assess the pollen quality?
Really no, but nurses are those, as we mentioned, those that ingest most of the pollen from the pollen reserves. They also are in charge of processing the raw pollen or the fresh pollen into the bee bread, which is some kind of first step in the food process that makes the food a bit more easily to digest. They consume big amounts of pollen, and then it uses such protein content to produce the jelly that share with other bees through prophylaxis. As we mentioned, we think that by consuming big amounts of pollen, they have the opportunity to evaluate whether this experience or this consumption leads to some distasteful, or as we said, malaise experience that affect not immediately the behavior of the bee, but 1 or 2 days after, when indeed these deterrent compounds can be detected or assessed. So exactly the process, or I don't know, but for sure, those components need to be ingested to give a good feedback about the quality of the resource. I think this is the way that nurse, by ingesting pollen, may have a full picture of the pollen composition. Otherwise, foragers that get access to the outmost layer of pollen, which might or might not reflect which is the content in the cytoplasm of the pollen grain. That is where most proteins and lipids are allocated.
Okay. Well, thank you very much again, Andrés, and congrats also to Emilia and Rocio on the work that they have been doing. And yeah, I hope to see many of you again next week for the next presentation, and I stop recording now.